y'all. So we are going to do our first little post-it lecture today. This is how we're going to be doing our post-its for the first section, or your first unit. We are in the biology book, and I am here on page 4. Okay, so what I'm going to do is I'm going to go through and show you my post-it notes and a little bit of background why they are important. All of these come from the reading. What I would like for you to do as you're following along is to pause the video as needed and add your own post-it notes so that you have something to study by, okay? And we're going to go through this entire section. So, what is science? The science of biology is what we're talking about. Biology is the study of life. This science can be applied to questions that can be observed, researched, investigated, or measured. Now, there is this nature of science idea. Science uses evidence to construct textual explanations and predictions of natural phenomena. It generates knowledge from people studying this topic. It's open to revision. It explains natural phenomena. It's a way of knowing. It assumes order and consistency. It's based on empirical evidence, and I've given you a little note down here. Empirical evidence is info that scientists generate by observation and data collection. Science, I have over here, does not prove things. As you continue gathering evidence, understanding improves, but you don't actually prove anything. Okay? So our goal here for science is to explain, understand, and predict things. Now, there is a generic scientific methodology. You've probably been taught before that there are specific steps in the scientific method that need to be followed exactly. Not everybody does experiments identically. They're done similarly, but everyone is a little bit different. Science is done a little bit differently by every scientist. Now, moving down here, we are going to move through generic steps of this method. So first you're going to ask questions. The question here is, why do marsh grasses grow to different heights in different places? They are focusing on one factor. Alright, focus on one factor, because when you look at more than one factor, it makes it hard to observe where changes are happening. Okay? Our next step is making a hypothesis. There is a difference between an inference and a hypothesis, and it explains that here. I didn't make a note for you, but an inference is a logical interpretation based on prior knowledge. A hypothesis is a tentative scientific explanation that can be tested. So a hypothesis is what you're actually going to be testing. So moving on. During the experimenting phase, you are going to have 3 different variables. You have a control group, which is what stays the same. You have a dependent variable. Dependent responds. It responds to the independent variable. And then your independent variable is going to be the one that the scientist changes. It's manipulated. So independent is manipulated by the scientist. Dependent responds to the independent. In this case, the control group, the one that stays the same, is the ground with no nitrogen being added. The nitrogen being added is the independent variable. And then over here, what responds, so grass growth, is your dependent variable. There are 2 types of data. One is quantitative. Quantitative, think quantity. It's a number. So in this case, it could be the number of plants in a pot or the length the height of the grass. Qualitative is quality, so that in this case is how healthy your grass is going to look. Now let me move back up for you. You will use statistics to analyze data, okay? Statistics doesn't necessarily have to be big scary math. It can be things as simple as mean, median, mode, and percent error. And we'll go through all of those at some point. After you have collected and analyzed your data, then you would create a conclusion. After your conclusion, you would go back and support or refute your hypothesis. This means either saying my hypothesis was correct or my hypothesis was not correct. And at that point, you're going to do more experiments. Repetition is key in science because the more that you do something, the more likely you are to have consistent results. You might have an anomaly one time, and if that's what you base your conclusion off of, then your data is not going to be correct. When you analyze data, you're going to display it in some way. So part of drawing conclusions comes from your data. In this case, they did a line graph. Line graphs are good for comparing values over time. We are now on page 9. Page 9. Here are a few qualities that are good scientist qualities. So good scientists are curious, they're skeptical, which means they question things. They're open-minded and they're creative. Okay? You'll see different figures throughout, which just expand on the figure on this previous page. So, technology is creating new opportunities for scientists, and ideas can arise in these ways. So, curiosity, observing things, being motivated by something, having a practical problem, and then again, technology. And scientists share ideas and they work together. A scientist is not somebody that is stuck in a lab by his or herself doing research constantly. Scientists work together in groups, sometimes huge groups, and they bounce ideas off of each other. And everyone has a specialty, and each scientist works on their specialty, and then they bring it all together to draw one big conclusion, and then they publish their work based off of what they've all contributed. One important thing to remember in science is that you have to avoid bias. So it is really strongly influenced by society, science is. And there are some limits because of cultural beliefs. There have been some sketchy things that have happened in the past that have contributed hugely to science, but people have questioned, because of cultural beliefs and morals, if those things should be used still because of how sketchy they can be. Okay? We're going to skip this section. We're going to come back to that. But I'm going to take you forward a little bit further. We're going to page 18. We're going to talk a little bit about measurements. And we'll come back to the other part later. This is a nice, short little section, okay? It is very, very important that measurements are accurate and that you use the same system around the world. America uses a little bit different system of measurement than other people. So we use things like pounds, inches, Fahrenheit. Most of the rest of the world does not use that. They use a different system. And it's important when you're doing science that you use that metric system, the one that everyone else uses in the world, so that if you do an experiment here and someone in Australia wants to replicate your experiment, you have the same measurements, you can do the same thing with the same factors. Here are common metric units. Let's see if I can get this to focus better for you. Hold on. There, okay. Common metric units. At some point you may have to do conversions, so we tend to use basic units or defi-centi milli. This chart, to me, is the easiest way to make conversions. So let's say that you have a basic unit. Let's say we're looking at 1 meter, and you want to convert it to centimeters. You would use your this chart and move your decimal place 1, 2 places to the right, and that would give you your new measurement. Now, when talking about measurements, you can describe them as being accurate or precise. Accuracy means the closeness to a known value. Precision means closeness of measurements to each other. So pretend that this is a bullseye, and the X in the middle, that just marks the middle. Precision means that measurements are close to each other. So if you see these 3 dots right up here, they're not in the center where the bullseye is, but they're close to each other, so those are precise. In this circle, the dots are right on the middle in the bullseye, so those are accurate. We know that the bullseye is the middle, and our dot is on top of that, so it is accurate. Now, if you look at this one, you have the bullseye in the middle, and dots spread all around it. This is neither precise nor accurate. You can have neither, or you can have both precision and accuracy. That is all for today. I will catch you again next time. We are going to talk about important characteristics of life.